Okay. So thanks everybody for coming. This is going to be a fMRI kind of workshop training series that we're going to do as part of the Neuroimaging Research Group. And we're just going to be kind of running through all the various stages that we go through in terms of generating and analyzing neuroimaging results and data. So this will be the first session, so I just wanted to kind of give an overall sense of what we'll be going over. So this first session will be focused a lot on the infrastructure that we use to set up our neuroimaging experiments, how we actually get the supercomputing cluster to run those, as well as kind of the incoming data formats. So I'll be going into a little bit more depth. I just wanted to let everybody know that I'm audio recording these sessions for later review, so that people that can't come here or that come later on can see all these processes as we go through. So the overall aim that I'm intending for these workshops and trainings is basically to review the current fMRI stream for pre-processing and initial analyses that we use in our lab. And while this will be kind of specific to the analytic approach that we're using in this case, the AFNI suite, I also want to kind of touch on some general principles that we have in our analyses. And the third and probably most important aspect of all of this is to answer questions or clarify anything that we find that's confusing about fMRI analyses, and anything, of course, that I say that's confusing. Please chime in and ask questions as we go. This is aimed at 4 sessions. If suddenly there's a developing need, we might be able to expand that. And after these sessions, we'll be, as Eric was stating earlier, moving to a broader format where we're doing more general review. So these are set up as in the next few weeks, barring the holiday breaks, to kind of review all these stages. Okay. So one big advantage that we have in our fMRI analyses is that we're able to utilize the Baker supercomputing cluster here to do to streamline a lot of analyses, and that also enables us to run a lot more data more quickly on a large number of subjects. So everything that I'm going to go through in these training sessions could be done on a single computer, but there's really a very big advantage to using a computing cluster. So I'll kind of go through these stages as we're going through. So the principal advantage of the high-performance computing cluster is that any user can come along and send really any number of analysis jobs to the cluster. And the big advantage there is all of the jobs that we send can be started and run at the same time. And that lets us essentially a task that would take 5 hours, if we did that serially, we would end up with, for 10 subjects, it's 50 hours. With the computing cluster, we're able to start them all at the same time in parallel, and that allows us to spend 5 hours running 10 jobs, and we've run 10 subjects. So this will become a lot more clear when we're moving into actually looking at the interface of the cluster. But the essential idea is that when you log in through a window, you're given access to this thing called QMaster. And that basically is a space for you to request resources. Then once you're logged in, you can, through a queue, request resources. And this is the naming that they have set up there. So you have these standard nodes here, and there's a lot of these, but they're not quite as fast as these high mem nodes. The high mem nodes have a lot more processing power, but there's only a few of them. In the process, when we're running all of our fMRI analyses, we tend to use these more plentiful nodes that we can run a large number of subjects. Though occasionally in harder jobs, we'll use one of these more powerful nodes. Sure.
So when I was using this HPO, they always advise me to use the node rather than HiMem. Is it very particular job they want us to use the HiMem, or it's something not for us? Like, for me, like they always recommended to go to the node and then start doing the.
Yeah. So so that's an important point. So to clarify, where you can request nodes on the computing cluster both for sending jobs in an automated fashion, but also if you want to hop onto the computing cluster and just view some results, that kind of thing, you can request one of these nodes interactively. This QMaster is it's sort of the bottleneck to the computing cluster. If too many people just did things in this node, it would make it really hard for everybody to use the resources. So yeah, that's one key thing, and I'll show those steps in a few minutes about how you log in to into a node to give us an example.
But Bonnie's point is that the high mem is reserved for more sort of special cases, and default would kind of be the regular nodes.
Yes, the default would definitely be the regular nodes. High mem is very useful for if you have something that you're doing that requires a lot of resources. Really computing intensive tasks, certain analyses, suites, like just take a ton of resources. Some MATLAB scripts run better in those. But generally, you want to use the nodes here that are slightly lower, just because these are really reserved for hard things. And it is important to note that we share this computing cluster with a lot of folks. And one of the major things, there's a lot of genetic studies that also use these same resources. So they have really hard stuff. Our stuff generally is a little more on the simple side. This isn't to say that these nodes here are bad. They're actually pretty fast, but there is that difference there. So there's, as Pavani got me started saying, there's kind of two ways you can do things on the computing cluster. I'm going to focus mostly on the queue system, but you can also use the computing cluster to just look at data and use its resources. So we'll see that part later. But the big thing that they use is there is there's a program and basically a set of systems that's installed on the cluster called Slurm Workload Manager. And basically what it does is it makes everything kind of more or less fair for people. So it sets up a system whereby you can send simple or complex jobs to a queue And as resources become available, whether it's right after you send it or at 2 a.m. The next day, it will go in and run what you asked it for, and it'll give you a lot of information about what happened or if something crashes. So that's kind of the next step that I want to focus on. So,
And I'll say that our analyses often will take a couple of days to run.
yes, yeah, we run into that a lot. So for this next step, I wanted to show the three main programs that we use to access the cluster. So there's this program. The one we use is WinSCP. This is a FTP or file transfer protocol client. Basically, what this does is it lets you upload and download files from the cluster. Now, when you first start with the cluster, they'll set you up with a username and then also some information about how you log in. And the key piece is the IP address that you log into. You're able to save all this so it won't work. And then you're able to log in with your, now with your typical hospital credentials, which is very nice.
Oh, is it? Okay.
Yeah, they did a recent change and synced up everything, so
Okay.
this will be a familiar password, so you don't have to update
Remember,
your Baker password periodically. So, for example, if I click login here, I'll get a window and a prompt for my password. I'll refrain from saying it out loud. And it'll give essentially a directory structure here. So on the left side will be your local or network resources. You can access the desktop or network folders. This is sort of your source, and this is the receiving end on the cluster side. There's an overall file system that we're logged into. Typically, when you start out, you'll be within this home folder, and there's a general directory for each of the various labs that you'll access. So there's even one for Hoskinson lab already set up. I'm going to use our particular lab. It's labeled based on the PI, I believe. So within this architecture, there's a series of folders. So this is my username. So typically, when I first go into the node, this is where, when I first log into the cluster, this is where it sends me. And this is just my personal scripts and things. This is actually mostly empty. Then there's generally a lab folder, and this is where all of our data folders that everyone is accessing will live. And generally that's divided into areas for our raw data as well as processed data. And we'll cover that all in a lot more depth. One additional feature, we'll call it, which is pretty positive, is every single folder and file on this file system has a set of permissions that says who gets to access these files. And you can allow people to look at the files, write to the files, or run programs. And that's really helpful, so you can have both common spaces, or if you're working on a particular project that you don't want anybody to accidentally delete or move files in, you're allowed to do that. So this is the structure for WinSCP. It works exactly like Windows does generally. You'll end up getting a status update when you move or download files that'll either pop up here or on your main screen that will tell you when your files are done transferring. So this is the main vehicle for us to move data back and forth from the cluster. The other thing happening is you are able to log in more directly to the nodes, not just viewing files on the cluster, but actually running things. Generally you do that through an application that works basically like a command prompt that allows you to securely log in to the computing cluster and run things. And that interface is doesn't have by default any graphical component to it. So it'll be just a black screen where you can type text. Fortunately, there's facility within the cluster to launch this program called Xming, and I'm just going to run it. And what happens is this just sits down here in your system tray, listening for information. So when you first set up your command prompt, the application we're using here is Extra Putty, but any variation of Putty will work. This is set up so that when you ask for something that's graphical, it'll actually make a window that you can interact with. So that allows you to do things like view your data and run programs or run something like MATLAB or Octave where you have to interact and type and give commands at various stages. So now that I've launched Xming down here, I'll go ahead and launch this application. This interface is where I do probably 90% of what I do on the cluster. In here I have this set up so that I can show you how you would log in here. You don't need to upgrade our data. So this is our host name we have here. So in this case, it's 10.101. And what's nice is you can also put your username here with an @ symbol, and then you don't have to type your username each time. Then the other key thing that you have to do so that Xming program can make a window, you just have to go under Connection, SSH, X11, and then check this box here that says Enable X11 forwarding. And once you do that, you can save all of these settings. I'm not going to bother with that right here, but you can save it so that essentially when you click on Extra Putty, it'll start logging you in and you can just access things quickly. This is just a restriction on this application. So the first thing it'll come up with here, I'm going to go ahead and increase our text size. So we can see everything. Okay, so when you first come into Putty, you get a screen that looks pretty much like this. So I specified my username. If I hadn't, it would have prompted me to. Using keyboard interactive authentication means you can type into the prompt. So if I type my password, you'll notice it doesn't actually show anything. That's like a safety feature. You do have to type your password correctly, however, into the prompt. Okay, here it'll tell you the last time you were logged in and from where. It gives you this little welcome screen and notes that the cluster is named after Sarah Josephine Baker, which is a very interesting person if you would read about her later. Here I am highlighting this strongly. So currently, and this may change later depending upon compliance, any... Yeah, yeah. I'm sure, but for the moment, eventually we'll maybe move in that direction. We'll see. But human subject data, that means any subjects you run that are humans, which will probably be all of them. It's important that no identified data end up on the cluster. And even if that does change, it's really useful to know about identification of study data, because more and more funding agencies and institutions are requiring you to share your, at least some form of your data. And so being aware that information like names and birthdays are stored by default is pretty important. And there's documentation here that you can look through. It's continually updated, and every time I send somebody through there, I discover something new that I wasn't aware of. And there's excellent support as well. I don't think I've waited longer than about 40 minutes to get a reply on any of my emails. As the cluster becomes used more, I'm sure that'll extend, but they're very responsive. So here's this stage here. So this is QMaster 01. There's actually a few nodes for this, but if you look over here, this is where that QMaster is coming from. So I moved through this pathway to get here. And it'll say what username I'm in. So as Bhavani was saying, one of the first things you want to do if you're going to actually do anything on the cluster other than send one job and log off, it's really important that you log into a different node. And the Slurm system helps organize and arrange that information. So I can use this command. S generally will be denoting something in Slurm, and this alloc for allocate. This allows you to request open resources. And if you can ask for a specific node, but if you just type this in, it'll give you access to the first available lowest resource node. Thankfully, they've seen fit to grant me a job allocation. So this is a job ID number. This will come up a lot later, but I want to draw your attention to it. It essentially tells you in the system what you're currently doing. It gives it a number, and this is really useful if something goes wrong. So it waited for a second, and then it said nodes, and then it gives this label, node 05 are ready for job. Other than being grammatically ambiguous, this means node 05 is the one that I'm looking for. So then you can use a command called SSH, and this is actually the same way you would log into QMaster. It's just the program does it for you. This is the program. You want to give it a switch or operator here of hyphen and then capital X. That tells it that it'll send X11 forwarding to Xming. So basically that's what lets you view a new screen when you're looking at your results. And then it needs one more piece of information without a dash, which is node 05. And I know that this is the node that I'm looking for because it told me earlier. So I'm allowed to access this one. So if I then push enter, occasionally it'll prompt you for a password. It'll also tell you the last time that you logged in again, just so you're aware. So the last time I used this node was the 31st of October. Not that that's that important. The big thing you'll notice here, though, and this is the way you can always double check if you're in the right place, is you have node 05. So now I'm not jamming up the works so that other people can't come in. Okay. When you're in here, commonly one of the first things you want to do is look at what's going on in our queue. So this is another Slurm command, SQ. I always have a hard time when I'm going over somebody's shoulder because they type SQ like this. But SQ, like the line, tells us where in line current jobs are. So if I type this, I'm going to get a whole stream of information. So way up at the top, SQ, you get these columns. Job ID is what job it is. Partition is where it's going on. Name is what the name of the job is that you decide. User is who's running it. ST is, here PD is pending, R is running. And then time tells you how long it's running, and nodes tells you how many nodes are being used, and it'll give you some information if it's not running. So remember here, our job right here was 1465960. So if I scroll down in here, you'll see here, this is the job I'm in right now, node 05. I've been in here for 3 minutes. I'm running right now because it's running here. And then it gives the job name bash, which essentially means that I'm using the shell. I'm not running a specific program that I made up. So that's how you can tell. And you can get some sense. So for example, there's a job over here that's been running for 21 hours. This one, somebody left this on. I know who this is too. This one's been on for 5 days, 19 hours. And so then these are different jobs in the queue. Usually there's either a lot of jobs or no jobs. And so when we have something that we want to run, we'll send it into Slurm. And basically you send a job into Slurm by, again, typing S for Slurm, and then batch, which means that you're sending a job, and then you type the name of the program that you want to run. Okay. So one neat thing about this interface is if you use the up or down cursors, there's a history of all of the things that you've tried to run, and there's a whole bunch of things in here. And you're also able to use this command called cd to change directories. Dot means you go up a directory. Dot means you're in the same directory. And what's nice about this is the tab button in this becomes your friend. So if I push tab, it says, okay, that's your current directory. If I push tab again, these are the directories that are one up from mine. So if I want to go into my lab directory, I can start pushing L for lab. If I push tab here again, it completes it for me. This saves a lot of time and potential for errors because it won't fill it in if it's not there. So if I go here again, here's all the directories that I could be in. If I want to then navigate into my raw data, this is this folder.
This is all using Unix language, right?
Right.
If you guys used it before?
Well,
No?
we got introduced to it.
Okay, yeah, so there are probably 15 or 20 commands that, I mean, it's
Yeah.
a lot of stuff going on, but there are probably 15 or 20 commands that can get you sort of
Right.
executing files and sort of navigating through structure.
Yeah.
Sort of thing. And you can
We-
find it on the internet.
yeah. Yeah, with a few basic commands, yeah. If you just search for bash commands, it'll start to tell you. Um, and yeah, you only need a few to really start accessing the jobs you'd like to run. So until I push enter, this doesn't actually change my directory, but if I do here, now I'm in piloting. One of those other commands that's really useful is ls, which just will list what's in that directory. Now you could go over in WinSCP and see this too, and navigate to the directories from that interface. So this here, over here, is just another way to show you the contents of the folder here. Generally, if I'm doing pretty basic things or running things, I will use the PuTTY interface if I need to move around files and see everything that's going on in a directory, particularly when we're talking about DICOM files, which I'll start talking about in a minute. It's a lot easier sometimes to see it this way. So cd and ls and these slurm commands are the very basics that you'll need to navigate around. There's a lot more commands that you can look at and that are useful for when you're running things, but if you have those basic commands, you'll be able to get around. And I'm happy, of course, at any time to talk and move through what some of those commands might look like. So the next thing that I wanted to talk about is to start us along the stream of how data moves in from we run a task at the scanner to now we're actually wanting to run our data for real, to ask questions about what's going on. And so when we first download our data from the scanner, and our scan staff is very, the scan tech all know about transferring our data off. We end up requesting our data de-identified for the earlier reason that we mentioned, and that we don't want to have any identified data on the cluster. And what we end up with is a huge number of files, and they follow a very specific naming scheme. You will get your whatever ID value you gave the person running the scan will end up here. There's a denotation for MR because you're running an MR scan. And then there's some additional information here about which MR scan it got culled in this case. And then there's this number here, and this is the series number, and that tells us the order that the files came in from the scanner. Basically, which thing was run first, which is really important when you have a bunch of files like this. Then this next one is the volume number, or sometimes we think about this as TRs. That's not totally accurate, but basically each round through the brain gets gets an individual number for this. And this extremely valuable set of characters is the date, time, and extensive information down to like almost, I think it's milliseconds or something, about this, about when this particular data volume was collected. The extension that the scanner generally gives us is this .ima extension at the end. One thing to note is that whatever, .text, .gif, .whatever file Linux doesn't care about that. It just does whatever you ask it to. So this is more for bookkeeping so that we know that it's this type of image. And so if we take a look at our DICOMs, so yeah, I even said one image here per TR. So basically, when you get your data, every single volume that you collect gets its own image file. And from this point, we generally want to convert it to some other file format that's easier for our analysis programs to read. So I wanted to show quickly what one of these raw DICOM files looks like. So if I scroll to the bottom here. So there's another useful command is called less. Basically what less does is it takes over the screen and you can use your cursor keys to read whatever you send it. So if I send it, if I can type, if I send it one of these image files. Notice tab was very helpful there in finding them. So this is the DICOM file from Series 63. It's the 119th volume, and it was gathered on 10/18/2017. So it gives you this warning. It's a binary file. Do you really want to look at it? Yeah. So this is the image file. So Les lets you kind of scroll through it. So if I go a really long way, you end up with a lot of information that's kind of gibberish, and eventually you get into straight binary, which is the raw data coming out of the scanner. But there's also a lot of tags here that give you information about, you know, what the protocol slice number was. So some of it's plain text and some of it isn't. The problem from the identification point of view is in here, if there's patient information, there's raw text that would say, oh, this is Whitney Matson, and he was born on this date, and he lives here sometimes even. So we want to make sure to not store any of that. Fortunately, if we anonymize, that all gets cold in here. And within here, there's information about like what protocol we ran, for example. So in here, there's the name of the sequence and what we ran. And actually, I'm having trouble finding it now, but in here also is the name that we gave the protocol. So this is all great, but this doesn't really translate for us into something that we can really run. So the next step that we want to do is to go through the process of converting all of these DICOM files into something that we can read. So what I'd like to do, I'm going to now switch temporarily over here to WinSCP, and we've got about 15 minutes left, so I'm going to start to show you this raw data script. So what I want to show you is a script that we wrote that was largely based on one study's file formatting. Initially, we were getting from the scanner, and then now has been converted into something more generalized that basically is it's a shortcut to running the commands to convert the raw those raw IMA files into the nifty format. In this case, it's the program AFNI that we use breaks that up into two files. One is a header file, and one is the actual binary data. So all that stuff that was text gets converted into our file header, and all of the binary data that happened at the end goes into the actual imaging information. So I'm going to pop into here. So this is one of our raw data study folders. Within this raw data, we have separate folders set up for individuals that we've run, and we have the output, as I was saying, the brick and head files for various DICOM series or various imaging series that we ran for our experiment. And then we also have a DICOMs folder that is quite large but contains all of the raw image files that we have for that whole session. So I have set up here a script. This is for this particular studies data, but it's a pretty general script and when it was written. And it ends with a .tcsh. So before, when I was typing commands in and it gave it that job name, I was accessing something called bash. That's the sort of the interpreter for the things that I type in. Generally, we tend to use this T shell, which has a few additional functions, and files that end in .py, which are Python scripts. So generally, those are the two languages that we use the most. So let me go ahead and open up this file. The start of any kind of file in Linux starts with this identifier, which basically says, hey, if you need to compile this file or if you need to know what it's written in, here's where you should look for what it should be interpreted as. So the reason for that is I gave it the extension .tcsh just so that for bookkeeping I could keep it straight. But you have to tell it what interpreter it actually is. So I don't need to type this last part. This tells somebody who's a random person who's opening up this file, okay, this is what this is written in. Every file contains these, or every well-written program contains comments. These pound signs or hashtags, if you must, denote something that's commented. So anything else that happens on that line gets ignored when the program's run. When we first get our data, we note where we start from. So this program can be run from anywhere, and at the end we go back to it. Then we have some lines that tell us which subject we want to run. So these set commands say, hey, I'm going to give you a variable. That variable is called subject, and then I'm going to set it equal to some value. So in this case, this script is set up right now to run our subject 63. And then top dir, this is where our files are. So you noticed before I was in raw data, SET1, that's the folder where the data is in. And then this is just the name of the DICOMs folder. So if we look back here for a second, in here, this is the name of this 63's data folder. This is where all their DICOMs are. Then what we end up doing is we get a whole set of series, and we note down what the order that we ran our protocol in, and then we have a series of commands where we say series dot the series number space and then what to call it. Here the $sign subject just means use the subject ID up here. So the fifth series here is called 01063 underscore ANAT. So that's our anatomical file. Here's our DTI file. Here's our face protocols, voice protocols, resting state, and then we have a reverse encoding thing at the end here. And these are basically just the series number. You know how it said MR, blah, 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 dot, a number, and then the volumes. That's where that comes from. And next time I'll show you guys an example of how to look up that information. But we wrote down all of these, so this queues up, it's essentially a key to what each series should get named. This is just notes for us later. So then what the program does is it goes into that DICOMS folder. And then it runs a command. So this is a command that you could type at the command prompt, but it's part of the AFNI software package. And what this command does is essentially it looks in the directory that you're in and looks in each of the DICOM files. So if we have DICOMs from all of these series in one big folder, what it goes in is it goes to each file and looks up what the series number was, and then it puts it in a directory. And so this calls the command star means run everything in that directory. So what it then does is it goes through and it moves all the different series into series folder called series.5, series.6, till it's sorted everything. Then what we do, and we can talk about loops a little later, but basically what this command does is it goes through all of the folders that it just made, and it goes in there and moves, where it just said series.5 before, to a new folder called, the subject ID underscore ANAT, or, series 13 gets moved to voice 1. Then it goes in there, and then within each of those newly renamed folders, it runs this command called daimon, which basically converts the DICOM data into that nifty format. And it basically names it according to what folder it's in. And then it just moves those resulting files back up to the base directory. So to show you a little bit of that, basically what it does is it takes these ID files and moves them into directories series.5. It renames them all, and then it puts them up one directory here. So the DICOMs kind of stay pristine, you're not messing with anything there, but it gives you these brick and head files that correspond to each of these series. As one additional step, we do a little extra conversion. We won't talk too much about DTI in this series, but within this script you can also convert it instead of to head and brick to a nifty file that contains information about other things that could be in the DICOM. And then it sends us back to the starting directory. So when we first get data back, the current system is that we get all of these DICOMs on a flash drive. So what we would do is we would plug our flash drive into our system. We would start up WinSCP. We would go to a subject's raw data folder. Here we would make a new folder for them. And then we would transfer all their DICOM data into a folder within that called DICOMs. So we would end up with a bunch here. Then we would make changes to this script so that we can move all of these and convert them. We would run the script, and then we would end up with our resulting files. To run a T-shell script, you would just type TCSH, so that's the program interpreter, and the name of our T-shell file, SCE T1 DICOM script. I'm making this up, but something like this. This is how you would prompt it, and then it would give you a bunch of information about did everything convert correctly. And then you end up with all of your resulting data files. And so this list of files right here, this is our starting point. These have been converted into brick and head files, but they're still our raw data. We haven't done anything to it. We just changed the file format. And so this ends up being the input that we take in when we start running our pre-processing steps. So the next time that we have one of these sessions, I'll go through the process of pre-processing that we go through with one of our fMRI functional tasks to show you how that goes and what our scripts look like for that. Then from there, I'll show a little bit of our quality control, and then if there's time in these sessions, maybe a little bit about level 2 analyses. So that's the basic overview of what I wanted to talk about in the first session. Okay, let me go ahead and stop this.